Hello and welcome. My name is Kathleen Ruddy. I'm the CEO of the St. Baldrick's Foundation. And thanks to you, I have the best job in the world because I get to work with heroes for kids with cancer every day. Our guest today is Dr. Crystal Makel. Dr. Makel is a physician scientist and immunologist. She is currently the Ernest and Amelia Gallo Family Professor of Pediatrics and Medicine at Stanford University. She's the founding director of the Stanford Center for Cancer Cell Therapy, the associate director of the Stanford Cancer Institute, leader of the Cancer Immunology and Immunotherapy Program, and director of the Parker Institute for Cancer Immunotherapy at Stanford. And if all those roles weren't ambitious enough, she's also co-leader of the St. Baldrick's Stand Up to Cancer Pediatric Cancer Dream Team, which she's here to talk about with us today. Dr. Makel, welcome.
Kathleen, it's great to be here. I'm so excited to spend some time talking about my favorite topic is bringing new therapies to children with cancer.
Thank you so much for your time today. So tell us about this dream team. When most people hear the term dream team, they think of Olympic basketball. So what is your dream team about and why is it so unique and impactful?
Well, first of all, the name is a little bit, I must say pediatricians tend to be humble folk. And we didn't give it the name, the Dream Team. It actually came from a Stand Up to Cancer funding model, which you know well, Kathleen, because they partnered with you back with St. Baldrick's back in 2013. But their model was let's find great athletes. We use the word athlete to discuss great scientists and great researchers. Let's find great athletes from the leading institutions working together on a problem and put them on a team together and give them funding to allow them to do things that they really could never imagine even doing in one institution in what we might call a silo. And so the Stand Up to Cancer group partnered with St. Baldrick's and said, let's put together a pediatric cancer dream team. And there was lots of competition for it. John Maris and myself put together a team of what we think were and are incredible athletes, folks that were committed to bringing immunotherapy into the armamentarium of childhood cancer treatments. And this was now almost a decade ago, at a time when immunotherapy was just beginning to be appreciated for its power. And so I think our vision was validated based upon all the success that's happened in the last, decade or so. So that's what the Dream Team is all about, a new way of doing science, bringing together very talented people from wonderful institutions and getting us all in a boat rowing together towards the same goal.
So when I got into this field about 20 plus years ago, scientists were talking then about the feeling that we had taken the traditional ways we fight cancer, being surgery, radiation, and chemotherapy, about as far as we could, or there was that concern that we were getting close, and they needed that fourth weapon with which to fight cancer. So immunotherapy looks like it's that fourth. Tell us more about that and about its potential and why it's maybe more humane than other types of weapons we fight cancer with.
Absolutely, Kathleen. So, when these things become real, in other words, when immunotherapy starts to have real benefit for real people across many institutions, many diseases, many trials, it can look like it came out of the blue. But of course it didn't. The idea that the immune system might be able to be harnessed to treat cancer started over a century ago. And it began with observations, things like patients that get bad infections sometimes would have their cancer regress or disappear. And for much of the 20th century, there was a loyal group of investigators. I actually got involved in cancer immunotherapy back in 1984. I remember very well reading an article published in the New England Journal of Medicine that demonstrated some interesting results with something called interleukin-2 in malignant melanoma. And so I was one of, a relatively small group that many thought were on the fringe. There were many of the more traditional cancer researchers who just thought that the immune system, that it was one of those things that, well, we tried that and it didn't work. But the truth is we didn't try it well. And our understanding of basic immunology just wasn't good enough. So over much of the 20th century, we learned so much about the human immune system, so that came, the 2000s, and again, everything started to really come together around 2010 is where we all saw the fruits of the labor. We really saw everything start to come to fruition. So I don't think there's any question now in the cancer field that immunotherapy is the fourth pillar. It has touched really every class of cancers you can think of, literally dozens of different cancer types. And honestly, every month it seems that there's another approval for immunotherapy for cancer. So it really is part of the armamentarium now, the fourth pillar.
That is incredibly exciting. It gives me goosebumps to hear that. But I'm curious about something. So my mother was treated with immunotherapy for non-Hodgkin's lymphoma about 21 years ago. Why did it take another 10 plus years or so to really get launched into pediatrics?
Well, so what your mother received probably was a drug called rituximab and an antibody, Kathleen, probably?
I think so.
Yeah. So, monoclonal antibodies were discovered it's sort of a synthetic in the 70s. In fact, two scientists got the Nobel Prize for that in the late 70s. But and if you go back to the mid 80s, about the time I was getting hooked on cancer immunotherapy, there was a lot of what came to be called hype, because cover of Time, cover of Newsweek, a new cure for cancer, the monoclonal antibody, the magic bullet. And while the idea was there, the concept was in place, reducing it to practice and getting around some of the nitty-gritty little details that got in the way took a while. And so it took from the 70s to about 1996 or so, when both Herceptin and rituximab were proven to be of benefit. Now, why didn't that? So those are monoclonal antibodies, and that really ushered in a time when monoclonal antibodies became part of the armamentarium. They didn't transform the landscape, though. They weren't good enough for every different kind of disease. And so Herceptin was for breast cancer, Rituxan was for non-Hodgkin's lymphoma, but it didn't spread out widely, and it didn't spread to pediatrics much. Why? Number one, they didn't work as single agents, so they had to be combined with other therapies. And so their impact was more modest. In children, we didn't have good targets for monoclonal antibodies. The target of Rituxan is expressed only on a very, very rare, only on a few lymphomas in children. It wasn't expressed, for instance, on leukemia. So there were just a lot of details that you have to, I think sometimes what happens in medical research, if there's a discovery that's made and it's so exciting and you hear about, New York Times, this discovery, it's going to cure cancer. And then years go by and people think, well, what happened? And oftentimes I think scientists too underestimate the time it takes to take a discovery to an invention. And the way you make a discovery is often like that. It's a few scientists working in a lab, eureka, I understand something. But an invention takes trial and error and iteration and optimization and going back and forth. It's more of a longer term process. And so I think that's what happened with, rituximab was a signal, but we didn't fully realize what immunotherapy could do with just rituximab.
So you talk about this, and you talk about some of those immunotherapy, those early agents needing a partner agent, if you will, to help the patient and to work together. in your dream team,
Yes.
you paired immunotherapy with genomics too. Why is that?
Yes.
And why was that pairing so inspired?
Yes. So, as I mentioned, much of the 20th century, the progress that was made that was so important was immunologists studying the immune system. So, if you're going to harness the immune system and hijack it or hack it or whatever you want to call it, when you take the immune system that has evolved to control viruses primarily, and you say, I'm going to use this army of killers to go after cancer, the only way you can do that successfully, number one, is you have to understand the immune system. So there are a lot of immunologists out there working hard trying to better understand the immune system. I'm one of those. I study T cells, day and night. But if you're going to train the immune system to go after tumors, you have to tell that immune system what to go after. The only way the immune system works is if it's got a target. Well, how are you going to find your target if you don't understand the cancer? So progress was only going to go so far if we only focused on the immune system. We also needed to focus on the cancer. Well, who focuses on the cancer? The genomicists. Cancer is a genetic disease. It is driven by abnormalities in the DNA that drive all these dysregulated processes that turn the cell from a healthy cell to a malignant one. Well, it's the cancer genomicists that have most of the knowledge and the databases and the understanding of what's the cancer look like and what might be on the cancer that we could target with the immune system. So what we found around 2013 when we were putting this together, John Maris is my partner here in this effort. And, John is a dyed-in-the-wool genomicist, brilliant scientist. And he thinks about the tumor all day long, while I think about the T cell all day long. So we really need that cross-fertilization to take the power of the immune system and begin to apply it in a very direct form toward specific targets in specific cancers. So that's why it was so important to bring together the genomicists with the immunotherapists so we could actually get some work done, make something that worked.
You said something really interesting. We talk about cancer as a genetic disease. We know that now. I don't know if that was always our understanding of cancer. It seems to me that at some point that revelation kind of became more clear to the field. And I think in adult cancers, you see more cancers that maybe are sparked by lifestyle or environmental causes. But those causes are often, or the cancer often occurs because they carry a genetic marker that when exposed to something will then trigger the cancer. Is that right?
It is. And Kathleen, what you've hit on there is exactly the reason why immunotherapies for children don't trickle down from adult therapies. Mm-hmm. So the real breakthrough in adult medicine, what made immunotherapy the fourth pillar in adult cancers, is antibodies, sort of like rituximab, except instead of the antibody going after the tumor, in this case, these were antibodies that have been developed to go after the immune system. And all those antibodies do is block an inhibitory signal. So it's like a double negative. If you block an inhibitor, you get a positive result, right? So these antibodies, called immune checkpoints, if you give them to many adults with cancers, adult cancers, especially melanoma, lung cancer, head and neck cancer, the list goes on, bladder cancer, kidney cancer, the immune system, it just goes after the tumor. Why? Because the adult cancers have all kinds of mutations. They have so many mutations that have accumulated through the life of the patient and through the exposure to these carcinogens. So some adult cancers have hundreds of mutations in each cancer that the immune system now sees as, oh, that's on the tumor and not on my body cells, so I'm going to go after it. Well, what about children's cancers? They don't. Children's cancers often have one or maybe 2, 3, 4 mutations. And the children haven't accumulated the mutations during the course of their lifetime. They haven't been exposed to these carcinogens. So how did they get cancer? Well, the children have a lot of what we would call stem cells are very young cells. And when a very rare but wrong mistake happens in one of these stem-like cells, boom, one mistake can take that cell all the way to cancer quickly. And so this is what happens a lot of times in children's cancers. You'll have one or two mistakes, and it dysregulates at a very early process. There aren't easy targets for the immune system to see. In fact, many times these cells look a lot like cells that were present in their fetal life. So from the immune system's point of view, they had to be tolerant to those. So we have to trick the immune system in a different way to go after children's cancers. We actually have to hack it. That's why we're doing things like gene therapy. We're putting synthetic receptors into our children's immune cells. The children's immune cells are tough as nails. I mean, they can go after and do what they need to do, but they aren't turned on by the tumor. So we have to trick the immune system to turn it on.
Fascinating. Just fascinating.
Yeah. Yeah.
So when your team was selected to receive the Dream Team grant, I think there was widespread confidence that the team could make progress in treating blood cancers like leukemia and lymphoma, but there was more skepticism that immunotherapy would be that viable new weapon against solid tumors. Why is that? And what have you learned now, 8 years or so later?
Yeah. No, Kathleen, this is one of the biggest questions that remains in the field. So there are many, so what we use, we spit it out, Chris. The receptors that I'm talking about, the synthetic biology that we use to hack the immune system, the best example of that are these chimeric antigen receptors. They aren't natural receptors. They are something that we use genetic engineering. We take part of an antibody, like part of the rituximab antibody, and we synthesize a molecule that attaches a signaling domain to that antibody. So then we use gene therapy, a defective virus, to put it into the T cells. I know it sounds like science fiction, but it's actually become very standardized. So when these immune cells get this receptor, they can attack whatever cell has that target. And back in 2010, the first proof of concept that these immune cells could work was observed. And that was in lymphoma, and it was also in childhood leukemia. We did some of these early studies, and the Children's Hospital of Philadelphia also did some of these early studies in childhood leukemia. Okay, now, so what about solid tumors? Well, the class of immunotherapies I mentioned in adults, the immune checkpoint inhibitors, where they just turn the brakes off and the T cells go after the tumor, those are happening in solid tumors. Those are happening in melanoma, lung cancer. But so far, there hasn't been the kind of high response rate in solid tumors using CAR T cells. So that's what's the unanswered question, not whether immunotherapy can work in solid tumors. We know it can. Immune checkpoint inhibition does. But can CAR T cells work in solid tumors? And the truth is, necessity is the mother of invention, right? This is often said. I think in the adult space, because they have immune checkpoint inhibitors, they haven't worked as hard on the CAR Ts. But in pediatrics, we know very well that the immune checkpoint inhibitors just don't work in most of our children's solid tumors. And so we have been really working hard to understand, why is it so hard to get a response in solid tumors with CAR Ts? And I think we've begun to crack the nut. We know that some of it is about the target. We have to pick the right targets. And some of it is about the suppressive milieu that the tumor sets up. And all of that is to say that once you understand the problem, you can engineer around it. You can create solutions. And so I think our team has made really remarkable advances in understanding this problem and in beginning now into the problem-solving stage. Why am I so confident? Well, neuroblastoma, of course, one of the dread diseases of childhood, children with high-risk neuroblastoma, while antibodies have had some benefit, our dinutuximab, given with chemotherapy, has definitely had some benefit. Many of the children with high-risk neuroblastoma still pass away. And we've started to now see that CAR T cells targeting neuroblastoma are having reproducible responses. This has been reported now out of a group in England and a group in France. I'm sorry, in Italy. And certainly, as part of our DREAM team, we are working very hard on that. In fact, we've just launched a clinical trial where we are testing these CARs in neuroblastoma in a multi-center trial in America and North America, Canada and America, that is sponsored by the NCI. So there really are signals. One of the group of our DREAM team have seen clear activity in rhabdomyosarcoma. And I think even more exciting, because we've had so little progress in pediatric brain tumors, we're beginning to see real significant responses using CAR T cells in pediatric brain tumors, especially diffuse intrinsic pontine glioma and diffuse midline glioma, some of the most devastating diseases. So, it wasn't something that happened on the first try. And so, there were some who said, well, we tried it and it didn't work. But there are those of us who said, well, we tried it. It didn't work. Now we have to understand why it didn't work. And now, as we iterate, we are now starting to see signals. So I remain confident, Kathleen, that CAR T cells are going to have an impact that is real and reproducible, meaningful for children with solid tumors and brain tumors. And so, just stay tuned. That's all I can say.
That's incredibly exciting too, because brain tumors as a group with the most lethal form of childhood cancers, and DIPG, which you just referred to, is uniformly fatal now. So the fact that you're seeing some ability to influence that disease is incredible, and I think gives so much hope to so many families. If you're in our audience or just joining us, we're speaking with Dr. Crystal Makel, co-leader of the St. Baldrick's Stand Up to Cancer Pediatric Cancer Dream Team. Dr. Makel, the progress made by the DREAM team is really unprecedented in pediatrics. And in fact, your team's success has eclipsed the other DREAM teams funded by Stand Up to Cancer. To what do you attribute this success?
Well, so, pediatrics is just such a different space when it comes to cancer medicine than adult medicine. There are the differences. I mean, I could talk all day about it. Part of it is that it's not driven by industry. It's driven by the academics. The innovation in pediatric cancer has to come from the investigators, the physician scientists, the clinicians at children's hospitals all over, really all over the world. And this is a very different model than adult oncology, where innovation there is largely coming from biotech. There may be some early work in the academic sphere. So part of it is, I think we already understood that it was on us to create new therapies. If we don't create new therapies, it's not going to happen in our arena. And so we are used to collaborating. We work with rare diseases, so we can't hunker down in silos. This isn't how pediatrics does business. So we were used to collaborating. We all understood the mission, and I think it was just a matter of the right people at the right time. And maybe it's that we understood that if we didn't do it, it wasn't going to happen.
Mm-hmm. The team is comprised of, I think, 10 institutions now in 2 countries. So talk a little bit more, if you will, about the collaboration and the value of that, and why working with multiple teams at different hospitals is so advantageous when tackling a problem as complex as cancer.
Absolutely. This is such an important point that really goes well beyond a discussion of cancer immunotherapy, cancer in general, just, biomedical progress and biomedical research. Collaboration, it doesn't matter what institution you're at. You can't have the expertise in every area. And sometimes, even at the most wonderful institutions, a certain myopia will set in. And that is you relate to what you see. And so you think about solving a problem the way the guy down the hall or the lady across campus is thinking about it. But when you bring people together from a fresh, a different ecosystem, you hear about new ideas, you hear about new technologies, you hear about, oh, yeah, well, we had this problem. There's this new technology that we're using that is a different way of doing it, or we're using the same technology, but we got different results. So, it's just the idea that getting a diversity of opinions in the same room, and forcing people to talk to one another, to present your results to one another early. By the time a paper is published, the truth is, it's already done. The work is already done. The place where real innovation happens is when the project is just starting. And that's when you want to have people around a table talking about results, discussing results before they're published. And so that's why, collaboration and building teams is so critical. You might think this would happen anyway, but really the way that medical research is funded through the NIH model doesn't breed it. It funds investigators. And much of the incentive is really to keep things close to your chest. And it's not the right way to certainly accelerate progress, especially when you're trying to develop new therapies. It might be fine for discovery, where you can kind of sit by yourself and come up with an idea with your small team, but you're not going to make new inventions by doing that.
Mm-hmm. So new inventions and new progress, you've talked about a number of diseases that the team is tackling. What kind of progress do you foresee, or what are you tackling next?
Yes. Yeah. So I think that there are two main areas. The first is that, we talked about cancer being a genetic disease, and there's no question that the genes drive it. But in some ways, I think that the advances in sequencing the human genome, it's very easy now to sequence DNA. So anything that's easy, it becomes, the go-to technology. But we're learning so much more about the proteome, which is what DNA does is it creates proteins. And proteins are the ways in which the actions of the cell are affected, are actually carried out. So a lot of effort on our pediatric cancer team now is focusing on proteomics. In other words, learning more about the proteins and learning how to quantify the proteins, how to pick the best targets, and just sequencing the DNA is not going to do that. So this is an area where collaboration has been great, because my group doesn't do a lot of proteomics, but the group in Vancouver does, and the group in Penn does, for instance. Another area is in synthetic biology. Synthetic biology is, as I mentioned, I think I used that word maybe when I talked about the CAR receptor, where it doesn't exist in nature. Mother Nature didn't come up with a CAR T cell. Keep in mind, the immune system did not evolve, for the most part, to treat cancer. It evolved to treat viruses, to keep infectious disease at bay. And so when we're trying to hack the immune system to treat cancer, we are asking it to do something that it doesn't normally do. We're asking it to get rid of billions and billions of cells that have a lot of defense mechanisms. So we're using synthetic biology. And what that means is we're creating ever more sophisticated pathways and receptors to train the cells to do what they need to do. Let me give you an example. Recently, we've been developing in our laboratory something called remote controlled CARs. Of course, we can use the car analogy. It goes on and on. But one of the problems with CAR T cells is, they're very potent. You put them into a patient, and then they can kind of activate the immune system, and some of the normal brakes are bypassed, and they can turn on too much, right? So we've all wanted to have a way to control the CAR T cells. We've put suicide switches in, but a suicide switch is kind of, just killing the cells you spend all the time to make isn't really soul satisfying. I mean, then, you've lost your chance to beat the cancer. You might have saved the patient, but, only, and leave the cancer behind. So we're now trying to get regulatable CARs that you can regulate this receptor so that If it's going gangbusters and the patient is doing well, all good. But if it starts to become too strong, you back off a little bit. And so we've created these remote controlled cars that can be turned on by taking a pill, and a small viral pill that's been FDA approved for many years and doesn't have any other effects. When we do that, one of the things we learned, surprise, surprise, is that not only can we prevent toxicity, but or reverse toxicity if it happens, but actually they work better. And we found out that the cars work better because what's happening is, as we give the pill every day, the cell gives a chance to rest. And so we've learned a lot about how to keep these cells going for the long run and prevent something called exhaustion. And when the cells get exhausted, they just kind of tire out. And by giving them periods of rest, we're able now to get more mileage in the long run. So we're working on synthetic biology. I could talk all day, Kathleen, about
It sounds fascinating and so incredible. There's so much innovation going on. And, from basically you're instructing the cells of when to sleep and when to be active and all of it. It's incredible. Now, all of this work that you're doing is incredible scientifically, but of course the outcome you're working for is the kids who need these treatments. So tell us about some of the kids who are alive today because of the Dream Team's efforts.
Absolutely. I think that the patients with leukemia, there are many dozens, hundreds now, children who have relapse leukemia, even back as 2010, was essentially relapsed refractory, not just first relapse, but multiple relapses. Very few of those children survive. Now, about half of the children will survive with CAR-Ts. And of course, I know that the community knows Emily Whitehead very well, who's now grown up and started her own advocacy efforts, and that's fun. And it's just fantastic. But there are many children like Emily, and Phineas has been one of your ambassadors, and of course we treated Phineas at the NIH, and I've known his family since we treated them. And I was just talking to his dad the other day, and he was talking about we treated Phineas when he was 4, and he's now in junior high, middle school. Being Phineas, he's always been a little ornery. I think that there are just so many kids like that who, shouldn't be, if you looked at the statistics, it wasn't going to happen for them. But now with the advent of immunotherapy, not only did they go into remission, but long-term, durable remissions. And like I said, we're at about 50% with leukemia. I think with the solid tumors, we still are nowhere near that. But there certainly have been children who are alive today that we didn't expect them to be alive with the advent of immunotherapy. So, let's hope that this is just the beginning, and there'll be many more in the future.
And to make sure that that wish comes true, how can people support the Dream Team's work?
Yes. So as everybody knows, the pandemic that's been COVID has been really devastating, especially for foundations. As we know, and I think, we mentioned the NIH is a wonderful source of research funding, but has not funded these kinds of collaborative, more innovative funding models, and certainly not the late translational work that the Dream Team has funded. So that the Dream Team has worked. So firstly and foremost, by supporting St. Baldrick's, who, we have a long-term commitment to one another, you would be supporting the Dream Team. And so I really, really hope that St. Baldrick's is able to come out on the other side of the COVID pandemic as strong as they were going in. Each of the pediatric Dream Team hospitals also are now matching funds. Myself at Lucille Packard Children's Hospital, or any other of the pediatric hospitals are also supporting the Dream Team effort. And so you could support your local, the one that's closest to you, perhaps it's a member of the Dream Team. But our foundations in the pediatric cancer space are so important, and there's none better really than the St. Baldrick's Foundation.
Well, thank you for that kind endorsement. And we certainly hope that this is just the beginning of what this incredible team can do. And I'm so grateful to you, Dr. Makel, for sharing your time with us today. You're obviously a very busy woman, but thank you so much for your co-leadership of the team and for sharing all the great results with us today.
Thank you, Kathleen, for this time. And thanks to all of you watching for all that you're doing to help our children get through these dread diseases. Be well.
And to our audience tuning in, if you'd like to learn more about the Dream Team's work, please visit the blog on the St. Baldrick's website and search by Dream Team. And be sure to tune in for our next episode this Friday, October 29th at 9 a.m. Pacific to hear Dr. Doug Hawkins talk about the critical work of the Children's Oncology Group. Bye for now.